For submitting an insect or arthropod for identification, follow these steps. First, collect the insect safely. Note that some insects and arthropods can bite or sting, so use a cup or container if necessary. Placing insects in the freezer for 24 hours is the easiest and most humane way to kill them. Then, prepare the insect for shipment in one of two ways. 1. You can place the insect in a vial or small container that is watertight and fill the container just enough to cover the insect with rubbing alcohol or ethanol. If a watertight container is not available, place the specimen in a sealable container and pack lightly with paper towel to prevent the insect from bouncing around during transport. When insects die, they are very fragile and can break if improperly packaged. If you need access to more materials for sample submission, consider contacting your local extension office. Next, place the container in a small zip bag, especially for containers filled with liquid. If the container is filled with liquid, add paper towel to the bag in case there are any leaks. Place any container in a padded shipping box or envelope for shipment. Next, fill out our submission form and mail in the form and payment if paying by cash or check with your sample. We offer two services for insect and arthropod specimens, a basic identification service and a DNA identification service. Most samples can be identified with a basic service that runs $10. We recommend selecting a basic service on the submission form, and if further DNA testing is necessary, we can inform you prior to running an invoice for the additional service as necessary. Once your sample arrives in the diagnostic lab, we will assign your sample a unique sample number and start your requested diagnostic service. This will always involve a broad screening of your sample by hand or under a microscope. For pathogen testing, we will place some of your sample in culturing medium to grow out any pathogens for easier viewing. Some samples require DNA extraction to identify specific pathogens and arthropods to the species level.